In this video, we're going to talk about proteins. Now, a protein is going to be a polymer that has more than 50 amino acid monomers. And the function of the protein is going to be determined by the shape of that protein. And the shape of the protein is going to be dictated by the attractive forces between the amino acid side chains. So here is an example of two amino acid side chains, and this is called an ionic bridge. We can also have a hydrophobic interaction, meaning between two nonpolar molecules, or we can have a hydrogen bond. Remember, a hydrogen bond is not an actual bond, it is a very strong attractive force. Another type of attractive force is going to be a disulfide bond. Have that sulfur in your side chain. The protein structures are going to dictate the shape of your protein, which then can dictate the function of your protein. So, a primary structure is where you just have the sequence of amino acids in a protein. So, here is an example of silk, and we have the different amino acids in silk. And the primary structure is just the different amino acid monomers bonded together. The secondary structure is going to be based on how many amino acid chains interact. And we can have two different types of interactions. We can have the pleated sheets. Wait, we are going to talk about the alpha helix first because that is the picture we have. This is the alpha helix, the alpha helix. A lot of times you'll just see alpha written as the Greek symbol alpha. And the alpha helix kind of circles around itself. And this is due to the interactions between the side chains. And generally, it's going to be that hydrogen bonding that causes these amino acids to wrap around each other. The alpha helix is mm, found in keratin, which is in our hair, and it has the alpha helix structure. We also have what are called beta pleated sheets. The beta pleated sheet happens when you have all of these different amino acids that are just kind of bonded together like so. And so here would be the view down. And silk proteins have pleated sheets in them. They have the beta pleated sheets. So both the alpha helix and the beta pleated sheets are going to be examples of secondary structures. So they get a little bit more complicated. The primary structure is when you have just the individual monomers bonded together. And then the secondary structures are going to be what they look like when you start taking those interactions of the side chains into account. Now, a tertiary structure is and alpha helix and an alpha helix together. They are going to be larger and have more of a three-dimensional shape. Hemoglobin is an example of a tertiary structure given right here. And you can see that there are a lot of areas of the alpha helix, but we also have areas of the beta pleated sheets. And then finally, we have a quaternary structure. Now, quaternary structure is essentially when you take a lot of those tertiary structures and combine them together. So it is the most complicated protein structures. So the full hemoglobin protein contains four separate protein molecules, as shown right here. Each one of these is a tertiary structure, but when you put them together, we get that quaternary structure.